Hello, let me introduce rodent cell line characterization. The rodent cell line characterization is based on the guidelines from the WHO ICH Pharmacopoeia of Chinese, European, and the United States. It is divided into 6 parts, including identity, purity, virus screen, rodent passenger, retrovirus, and bovine and porcine virus. Firstly, for the cell line identification, we refer to the ATCC guidelines and use the Sanger sequencing methods to sequence the COVID gene, and then compare the sequence results with the BOLD database to determine the species of origin. This method offers higher sensitivity and specificity compared to the isoenzyme analysis method. After confirming the identity of the cell line, microbiome testing is required, which includes the sterility test, mycoplasma detection, and the mycobacterium detection. Bacteria and food contamination are detected using direct inoculation and the mobile filtration method. We also developed a rapid sterility test. Mycoplasma detection employs culture method and indicator cell techniques referred to European pharmacopoeia. Mycobacterium detection used a culture-based method. We also developed a rapid mycobacterium detection based on qPCR. For the detection of bacteria and food contaminations, the test is conducted in a validated ISO Level 5 environment within an isolator. In accordance with the requirements of the European Pharmacopoeia, it is necessary to test 1% of the cell bank or not less than 2 vials of the cell bank. There are two methods used: direct inoculation and mobile filtration. Typically, cell samples are tested using the direct inoculation method, while UBH samples are tested using the mobile filtration method. Mycoplasma test employs culture methods and indicator cell methods. The culture method involves culture in liquid medium, followed by inoculation onto solid medium for further cultivation. The indicator cell method uses virus cells as indicator cells, which are cultured for 3 days and then strained with culture dye. In addition, we have developed a mycoplasma detection assay based on the qPCR method, which is capable of detection over 200 species of mycoplasma within 2 hours. Mycobacterial tests use the culture method with a reference to the European Pharmacopoeia 2.6.2 bacterium for a 56-day incubation period for detection. After confirming that there is no microbial contamination in the test articles, it is necessary to eliminate the risk of viral contamination, including both retrovirus and adventitious viral agents. Before testing, the possibility of the viral contamination must be considered. For example, whether cells obtained from infected animals and whether the cell bank has been exposed to the animal origin materials. Additionally, comprehensive risk assessment should be conducted, for example, historical and origin of cell line and the animal origin material used in process. For adventitious viral screen, in vitro and in vivo methods are used. For the in vitro adventitious viral screen, a 3-indicator cell method is used, employing MRC5 viral and CHO cells as indicator cells. After 14 days of culture with regular CPE examination, a blind passenger is performed and then cultured for an additional 14 days onto fresh indicator cells. Throughout this process, the appearance of CPE is observed. If CPE is detected, hemagglutination and hematosuppression tests are conducted accordingly. For the in vivo adventitious viral screen, 3 model animals are used, including suckling mice, adult mice, and embryonated eggs. For suckling mice, the test articles are injected into 10 suckling mice each. After 14 days observation, then the liver, lung, kidney, and heart tissues homogenization will be injected into 5 mice. Continuous observation for 14 days. For adult mice, the test articles are injected into 10 adult mice and continuous observation for 21 days. For embryonic eggs, the test articles are injected into yolk cavity and allantoic cavity. For the detection of retrovirus, 3 methods are used: transmission electron microscopy, quantitative fluorescence product enhanced reverse transcriptase, and infectivity assays. TEM detects the presence of virus particles through observation. CurePERT is a highly sensitive and universal method for detecting retrovirus. If CurePERT results are positive, an indicator cell assay is subsequently performed to determine the infectivity of the virus. In addition, according to the recommendation of ICH-Q5AR2, we employ mouse and hamster antibody production tests to detect specific murine virus. The test articles are inoculated into mice or hamster, followed by collection of blood from the tame mice. ELISA methods are then utilized to identify specific urine virus, for example, hantavirus and centanvirus. In response to the European Union's 3 Is replacement, reduction, and refinement initiative, we have also developed a series of qPCR-based methods to detect murine virus capable of infecting human or human cells. If animal-derived components are used in the manufacturing process, it is also necessary to test for bovine and porcine virus. We use 2 methods. One employs the viral indicator cell method combined with ELISA to detect specific bovine and porcine virus. For viruses that cannot be detected by the indicator cell method, we have developed a qPCR-based method for porcine and bovine circovirus and hepatitis type E virus.